Hello students, myself Dr. Pallavi Bhale and I am going to teach you the applications of plant tissue culture in today's topic of Pharmacognosy BP 405T. So today we will be continuing that we have studied everything about plant tissue culture, from how the explants are selected, from how we decide that what will be our nutrient medium, how we will increase the secondary metabolite production, but how is it useful in basically in the pharmaceuticals that we are going to discuss in today's class. So in short, if I see, there are few points which we can you can say classify, and then we will look at the various examples in the pharmaceutical sector where a breakthrough has been achieved due to the plant tissue culture. So plant tissue culture technology has been used in almost all the fields of biosciences. The desirable products produced by the plant tissue cultures are as diversified as the industry itself. Now here if we start with the applications, it has been seen that it increases the production of phytopharmaceuticals. It is also used to perform the biochemical conversions in the cells. It is used to do the clonal propagation or the micro propagation, and an extension to this clonal propagation, it also does the soma clonal propagation. We will be discussing that too. And then lastly, the production of immobilized plant cells, which are used for further different types of processes. So starting with the phytopharmaceuticals production. So what do you understand by phytopharmaceuticals first of all? See, those chemical entities which are isolated from the plants and in today's time are used as API, active pharmaceutical ingredients. For example, atropine. You know that atropine eye drops are available in the market. So atropine is what? It is obtained from Atropa belladonna, but it is available in the market in the form of a formulation which is completely a allopathic formulation, completely a modern medicine, right. So there are many ingredients which are in present date available as modern medicine, like vincristine, vinblastin, you have reserpine. There are many of them which are available in market in their active pharmaceutical ingredient form or in their isolated purified form. Such ingredients are known as the phytopharmaceuticals. So it was first of all in this plant tissue culture history, it was a scientist named Weinstein and his group that in 1959 they started studying the agave plant. There is a agave plant and they started studying this agave plant for the production of steroids using the plant tissue culture. The plants, they also synthesize a number of steroids, like you have studied about the steroidal saponins, the sterolic compounds, cardiac glycosides, they all have a steroidal ring in them. Okay. So plants are easily able to synthesize the steroids. We humans also require many drugs which require the basic steroidal ring, like the androgens, testosterone, you have estrogen, progesterone, corticosteroids. All these steroidal hormones, they have the same basic ring structure. Now if we require a source of these steroids, it is very difficult to synthesize the steroid inside the lab itself. So it is better if we have a source or a natural source where the plant it is going to synthesize this steroidal ring. So it has been researched since ages that which are the plants which can give us the steroidal ring easily, and agave is one of them which Weinstein and group studied, and they could successfully see that it started synthesizing steroid at a larger scale. Then further, Diascorea was also studied. Again, Diascorea is a drug which contains diosgenin. Diosgenin is again a steroidal drug, and thus Diascorea has been reported to contain industrially useful steroids by 1966, and it was Call who reported that we can easily increase the production of diosgenin by about 1.2% by doing the tissue culture of Diascorea sylvatica. So they did the plant tissue culture of which plant? Diascorea sylvatica, and they saw that diascoria could easily increase, we could easily increase the percentage of the diosgenin inside the plants by doing it inside the lab. So, this is one, you can say, important finding or application of plant tissue culture. Even the calyces as well as the suspension cultures, they are also capable of synthesizing secondary metabolites, and the yields are comparable to the intact plant. If we compare that, if we have done the callus culture and suspension culture of a plant inside the lab, it has been observed that it is not possible at times, we have to do cultivation at a very large scale if we require a lot of drug, and we do not have that much space to cultivate, as well as it takes a lot of time. If we see, if we are planting a seed and we wait for it to grow, then it matures, some trees take many years to grow. So, it becomes a very long process to get the active pharmaceutical ingredients. But if we start doing callus production and the callus itself is enough for us to give us our main chemical constituent, then why do we have to wait for that time? We can do it at a very fast rate. Secondly, it is also not required that we have to do a cultivation at a big land area, or hectares of land are getting used for a particular cultivation with a very less produce. It is more feasible if inside a lab itself we are getting more amount of drug. So, that is the benefit of doing plant tissue culture. The second application of plant tissue culture is that we can make many of the biotransformations or biochemical conversions of the main active chemical constituents of the plants easily by doing plant tissue culture. So, here what happens is that we are transforming or doing a chemical change in the chemical molecule inside a biological system or our plant. So, one of the examples for this which has been performed is that a plant named Digitalis lanata. Now, Digitalis lanata is well known for its effect on the heart. It is a cardiotonic drug. So, it has been seen that Digitalis lanata, it contains a component named beta methyl digitoxin, and when we did the plant tissue culture of this Digitalis lanata, it was seen that we could easily do many reactions inside the callus, like we could go for hydroxylation, addition of hydroxy group, acetylation, addition of acetyl group, or glycosylation, addition of a glycosidyl group. So, we could add these components inside the particular inside the chemical constituents present inside these callus of the Digitalis lanata easily, and therefore, we could produce the ingredients which are of our choice. For example, that it was reported that in Digitalis lanata, if we used the strain 291, this strain could convert beta methyl digitoxin into beta methyl digoxin, and it has been observed that digoxin is more effective compared to digitoxin because of its less cumulative effect inside the body. Digitoxin has a more cumulative effect inside the body compared to digoxin, and therefore, it is of more benefit to us compared to digitoxin, and by plant tissue culture we could easily convert beta methyl digitoxin to beta methyl digoxin. Then another study said that when the cell suspension culture of Strophanthus gratus was done, we could easily convert or we could make biochemical conversions in the molecule of digitoxygenin. So, digitoxygenin, it is the A glycone of the cardiac glycoside digitoxin. So, we could convert digitoxygenin into digitoxin, and digitoxin, which is a secondary glycoside, it could be used as a drug or as a API for various cardiac issues. Then in the third example, it has been observed that the monoterpenes are those compounds which have the C10H16 basic structure. So, in monoterpenes, we could easily do the bioconversion in the mentha cell culture. So, mentha is a spear, so it is basically a peppermint plant, and in peppermint plant it has been seen that it is a very rich source of menthol. Menthol is the main source, but other than menthol it contains many other drugs, for example menthone, pulgen, etc. So, when these monoterpenes were treated, or where certain chemical reactions were done in this mentha, which was being grown through a plant tissue culture, there it was seen that menthone could be easily be converted to neomenthol, while pulgen could be easily converted to isomenthone inside the lab. Further, another drug which is Podophyllum peltatum, which is an anti-cancer drug. Okay, it is an anti-cancer drug, and its semi-continuous culture was done. It was seen that it led to the synthetic production of dibenzyl butanolides, which could be converted to lignans, which is helpful in the synthesis of etoposide. So, etoposide, it is a semi-synthetic anti-cancer agent, and the podophyllum, it gives the basic raw material for the synthesis of etoposide. So, when we did the semi-continuous culture of this particular Podophyllum peltatum, and inside the lab there were certain biotransformic conversions. So, we could easily convert the dibenzyl butanolides inside the plant into a lignan, and this lignan was better precursor for the synthesis of etoposide. So, that was the benefit of doing the culturing of Podophyllum peltatum. Then in some tissue culture, stereospecific biotransformation has been reported. What is stereospecific biotransformation? When the stereoisomers of the particular compounds can be converted inside the plant tissue culture. So, it was seen that we could isolate the optically active compounds from racemic mixtures. For example, in the cell culture of Nicotiana tabacum, selectively it was seen that the hydrolysis of R configuration form of the monoterpenes was being done, like in bornyl acetate and isobornyl acetate. So, here only the R configuration was getting hydrolyzed, because of which we could only get the S configuration, and we required only the S configuration. So, it became easier to get the only the S configuration from the plant, rather than getting the racemic mixture and then separating them into two different configurations. So, we can do the stereochemical variations also using plant tissue culture. So, apart from all these above mentioned reactions, it has been seen that there are different biochemical conversions, like we can do the saponification, esterification, epoxidation, oxidation, methylation and isomerization of these different chemical constituents of the plant drug. So, everything is possible inside the plant tissue culture system. The third application here is that the clonal propagation. So, clonal propagation, it is also known as micropropagation. Now, you know what is cloning. Cloning means when you take a particular thing and you start preparing a copy of that particular thing, that is a cloning. Okay. Now, when you eat fennel, fennel is soft. Okay. So, when we bring soft at our home, you must have observed that the soft is not of a same variety whenever you get. Sometimes it is very thin, sometimes it is very big in size, sometimes it is bitter in taste, sometimes it is sweet in taste, sometimes it is very aromatic in taste. So, you can understand that fennel is a thing which where the species to species variation, or you can say area to area variation, like if you get fennel from Rajasthan and if you get fennel from MP, the type of the quality of both the states will be very different. So, this varieties of the fennel are very easily they can be changed. So, now if a particular industry, like a company which is producing any kind of toothpaste where he is adding fennel oil, now if he requires the same type of fennel source, what will he do? He needs to have the fennel of the same quality. So, for having the phenol of the same quality, what is the best possible technique which needs to be followed? You have to do the cloning of the phenol. You keep on cloning the phenol. So, you will have the same copy coming again and again and again, and there won't be any variations in the quality of the phenol which you require. Okay. So, this is the basic purpose of doing clonal propagation. So, here clonal propagation is the technique to produce the entire plant from a single individual by asexual reproduction. We are not making any change in the cellular properties of the cell. This fact can be commercially utilized to produce high yielding crops of desirable characters in a short period of time, which otherwise show variation when grown using seeds. So, we can avoid that variations which are there when we use it, grow it through using seeds. For example, Funiculum vulgare, that is fennel or soft, shows wide variation in yield and composition of the volatile oil, and by this technique it has been reported to have uniform clones of fennel with narrow variations in the volatile oil compositions in comparison to the asexual cultivation. So, one of the best example for clonal propagation is fennel. Now, again extension to this particular clonal propagation is soma clonal variation. Now, what happens is that when we are doing cloning, so there is a asexual reproduction. So, the types of the younger ones which are formed, they should have the same genetic makeup as the parent plant. There should not be any variation. However, it is a biological system, and when there is a biological system, what happens is that if you are producing around 100 plants or 100 young ones, there is a probability that there will be 1% chance that one plant will be of a different property, because it is a biological system. So, there comes a variation. So, 1% variation is possible when we are doing the clonal propagation, and that variation is known as a soma clonal variation. Okay. So, in clonal propagation, clones are produced from tissue culture with uniform characters. Okay. They have a uniform characters, but few clones may show variations among the population of the clones which are not present in the parent cells. This formation of variant clones from the cultured tissue is called soma clonal variations. Okay. Now, this type of variation can again be of two types. It can be a desirable variant, it can be a undesirable variant. Okay. There are two things, it can be a good variant, it can be a bad variant. Good variant is what? Which will be of our benefit. It is giving up a added property to the plant, or it can be of undesirable variation, whether it is giving us a low quality of the chemical constituents or low quality of yield. There are two possibilities which are there. So, what is our benefit? We have to focus on those variants which are showing the desirable variations, and we can use these properties to develop our next crops. Okay. So, this is giving us a chance, a hope that we can have a variation with a better quality from the variety which we have been using for clonal propagation. So, desirable variants can be used for improvement of crops, and the clone showing high productivity can be used for the commercial purposes. We can start using it for commercial purposes if we get a variant with a desirable property. So, you have seen many kind of the plants, they have a very different type of property, like there are many fruits which we eat. Earlier they used to have seeds in them, but gradually these seeds have been lost. So, there are different reasons for that. There are certain techniques which have been applied, after which we could prepare those desirable properties in these plants. But yes, in present times, whatever fruits, vegetables, medicinal herbs, the natural plants which we are consuming for our purpose, generally they have been modified a lot by the humans, and there are many variations which have been induced by humans, and after which we are continuing to use it. For example, you must have learnt in your unit 2 about polyploidy. So, polyploidy is the technique where you increase the number of the chromosomes. So, after polyploidy, we are in what happens that there is an increased set of chromosomes inside the plants, but there are many plants in today itself which are having this polyploids, like the wheat we use, the wheat which we make flour from. It is generally I can say that 80% of the wheat we are using today, it is having the polyploids in it, and they are not the original set of chromosomes. Then we come to the next topic, that is the immobilization of the plant cells. Now, what is the benefit of immobilizing the plant cell? Now, when we immobilize the plant cell, we can use the immobilized plant cell to perform any kind of chemical reaction or activity which we want. So, that is the benefit of immobilization. So, immobilization of plant cell or enzymes has increased the utility of plant cell biotechnology for production of pharmaceuticals. The plant cells, they can be immobilized by using matrix like the substances like alginates, polyacrylamides, agarose, polyurethane fibers. These substances can be used and they can fix the plant cell at a particular place. So, once they are fixed at a particular place, we can perform any kind of processes or methods can be adopted on these plant cells. The immobilized plant cells can be utilized in the same way as the immobilized enzymes to effect the different reactions. Immobilized plant cell systems may be used for bioconversions, such as we can convert the codenone to codenine, digitoxin to digoxin, or we can also do the synthesis of various precursors of different chemical constituents. We can increase the production of agmelicin from tryptamine and cycloganin. Now, these are few chemical constituents which are present in different plants, like codenone and codenine are present in the opium alkaloids or the opium, which we call it afim. Digitoxin and digoxin is present in digitalis plant, which is a cardiotonic drug. Then this agmelicin, it is generally present in plants like vinca or vasakha or rauwolfia, which are the Apocynaceae family plants, and you will find that they all contain agmelicin in them. Agmelicin is synthesized by two substances, that is tryptamine and cycloganin, which are the intermediates of the synthesis from the amino acids. So, amino acids are getting converted to tryptamine and cycloganin. From amino acids and cycloganin, we have the synthesis of various alkaloids. One of them is agmelicin. The suspension culture of Anisodus tanguticus has been reported to convert hyoscyamin to anisodamine. So, hyoscyamin can be converted to anisodamine in Anisodus tanguticus by using the suspension culture. Further, anisodamine can be converted into a scopolamine. So, scopolamine is again a solanaceous alkaloid. It can be formed from anisodamine. The biotransformation reactions such as glycosylations, hydroxylation, acetylation, demethylation have been successfully implemented in the immobilized cell systems. It has been seen we can do the hydroxylation or glycolization of cardiac glycosides in Digitalis lanata and Docus carota plants. So, these plants, the Digitalis lanata and Docus carota, they have been studied a lot because they give us the cardiac glycosides, and in them we can do the hydroxylation where we can convert digitoxin into digoxin, as well as glycolization where we can convert the aglycones into secondary or primary type of glycosides or the cardiac glycosides using the immobilized plant cell. Then finally, I can say that immobilized plant cell can be used for tracing the biosynthetic pathways of secondary metabolites and can also be used for carrying out biotransformation and biochemical reactions. So, basically these 2 points, like when we are studying any kind of plant, we have to understand that how are the secondary metabolites being synthesized inside the plant. To understand that, we normally study the biosynthetic pathways present inside the plant. For studying those, it becomes very easy if we are able to immobilize the cell at that particular stage, and we are able to understand that what are the chemical reactions going inside the plant by simply radio labeling those chemical constituents. Like, what is radio labeling? We are replacing the carbon 12 with carbon 14. We are replacing the hydrogen, which is normal hydrogen, with deuterium or tritium, and because it becomes radio labeled, we will understand that what are the chemical reactions taking place inside the plant cell. So, that is how we can understand the biosynthetic pathway, and even we can start inputting certain chemicals from outside to increase the biotransformation or biochemical reactions of our plant cells. So, these all processes are possible by immobilization of the plant cells. So, with this we come to an end of all the applications of the plant tissue culture. We have studied about it increases the production of the phytopharmaceuticals, we can do the clonal micropropagation of the cells, we can do the biochemical conversions inside the plant cells, and we can immobilize the plant cell and perform whatever type of reactions or productions which we require. So, everything is possible by using this technique of plant tissue culture. So, with this we come to the end of the plant tissue culture topic. Now, in the next lecture we will be continuing with the edible vaccines that we will be discussing. Thank you.